With regard to the article titled Senescence Reprogramming by TIMP1 Deficiency Promotes Prostate Cancer Metastasis, we will be presenting on figures 1B through E. So just to recap on the big paper question and figure 1A, the question proposed is, does PTEN loss induced senescence lack factors that cause progression and or have factors that inhibit metastasis? So figure 1A here shows a sample of 100 mice over the course of 100 weeks and their survival percentage with regard to time, each with different specific genes that target tumor progression. And it shows the rate of cancer metastasis with a decrease in lifespan. So the PTEN double positive, shown by the blue line, TIMP1 double negative, and PTEN double negative, which are not able to be seen because they're under the blue line, all experienced no decrease in survival percentage over the course of the 100 weeks. The PTEN double negative with TIMP1 mice experienced a decrease in survival percentage starting as early as about 35 to 40 weeks and tapering off a little bit after 80 weeks. That's shown by the orange line there. PTEN double negative with TRP53 double negative showed a more drastic increase starting at about 30 weeks and sharply declining, ending at about 35 weeks. And finally, the PTEN double negative with TRP53 with TIMP1 as well showed the most drastic decrease in survival percentage over the course of weeks, starting as early as about 20 weeks and ending at about 30. So to conclude from this figure, it was found that TIMP1 is the most upregulated factor in figure 1A here, and that the hypothesis that was deduced from this figure was that TIMP1 could prevent senescent cells from triggering metastasis in PTEN tumors and even act as an inhibitory factor. So now in addressing figures 1B through E, the figure objective is senescent versus non-senescent tumors and the effect that particular drugs have on them under certain stains and tests. So I just covered figure 1A. Here looking at figure 1B, it addresses senescent versus non-senescent tumors with regard to a bunch of different stains and tests. And the question proposed for that figure would be, can TIMP1 reduce cancers with and without senescent tumors? Figure 1C here is a bar graph showing drug stains and how the individual genes are affected, showing TIMP1 and PTEN and TRP53 with regard to certain stains in a bar graph form. Figure 1D here shows senescent versus non-senescent tumors again with a few different stains and age-matched mice. And the question proposed there is, can TIMP1 have a negative effect on cell decision? And once again, that's shown by the drug stains. And finally, figure 1E is P16 and CDH2 displaying metastasis from specific tumors and to find what genes specifically promote this. So in the article, the specific title of figure 1 is PTEN TIMP1 null tumors displayed tumor progression and metastasis with decreased lifespan in mice. So that's just a general overview of the entire figure. So specifically in the approach, the main steps included here would be first to gather test subjects, which in this case are male mice since they are the only sex of mice with a prostate. And these are prostate-specific mice that are crossed with TIMP null mice. And this is important because it sets up the experiment and gives us our test subjects that the entire experiment is based on. The second step would be to confirm gene expression with immunohistochemistry to ensure that the experiment being conducted will be correct and all of the test subjects are displaying the genes that they're supposed to be displaying and we have them correctly labeled. And the final major step would be to obtain the samples and tests so that we can get actual factual data and analyze the progression of tumor cells under the different stains and conduct our experiment. Okay, now figure 1b specifically here. So just to give some background information, these are the different stains here on the left side, and these two columns on the left are senescent tumors compared to the two columns on the right, which include non-senescent tumors. This column here is P10 double negative mice. This here in the second column is P10 double negative with TIMP1. This is P10 double negative with TRP53. And here under, I don't know if you can see that with my face there, is P10 double negative with TRP53 and TIMP1. So this H&E stain, which is hematoxylin and eosin stain, shows all of the cell structures in cells, which allows us to see basic cell function and specifically the nucleuses of particular cells. It's shown by the purple dots, which are shown, as you can see, in each of these here. The TIMP1 stain is an inhibitory molecule that regulates metalloproteinases, which are responsible for breaking down enzymes, and it is shown with brown marks, which you can see in the P10 double negative and the P10 with TRP53, more brown in each of these, with these two not showing as much. The Ki67 stain shows cell proliferation and is essential for the sheath that surrounds chromosomes in particular. So the more of this protein that's found, the faster they're dividing to form new cells. And this is shown with the brown dots, with these two on the right showing the most brown dots, particularly the P10 with TRP53 showing the most in the darkest spots. The beta-gal staining, also known as lactase, is something that breaks down lactose to produce energy and carbons, determine the transfection efficiency and expression of the particular LAC-Z gene. This is also used as a marker of tumor progression, and this is shown with the blue markings, which are seen in the P10 double negative and the P10 with TIMP1, with the P10 double negative displaying the most blue, and neither of these on the right here showing really any blue over there. The P16 protein stain here is a protein that slows down cell division, so it is a tumor suppressor protein, and it is shown with brown dots, which can be seen across the board here in all of these, particularly the most in the two senescent tumors, which are the P10 double negative and the P10 with TIMP1. And then the final stain here is the pan-CK CDH2 and DAPI, which are used to determine the total number of nuclei and assess gross cell morphology. And very clearly you can see the most lit up stain from the P10 with TIMP1 stain there. And then the conclusions of this figure show that TIMP1 genetic in activation in senescent tumors, but is not found in non-senescent tumors, resulting in metastasis. So here's figure 1C. Each of these bar graphs on the right is the percent of a stain on a different combination of genes. And as the percent increases, the rate in which the cells divide increases. So an increase in percent is not something you want for each gene. So over here, I've highlighted what stains are being used, which also is displayed on the left of the graph. And down here, according to the positives and the negatives, it shows what genes are present. And for example, CDH2, since this one's kind of an outlier compared to the rest of them, it only interacts with the metastatic cells. So it has a very high rate of cell division, and only TRP53 is present. So that makes sense because in previous slides and the next one, TRP53 is not a good gene to have by itself in terms of slowing down the rate of cell division.
I'm gonna go over figure 1D. So figure 1D shows two types of progressed tumors in the lymph nodes of mice. We have senescent tumors and non-senescent tumors. Both tumors are dated at 34 weeks of age as far as the mice are concerned. So all the mice in each of these stains were 34 weeks of age. The senescent tumors have three different types of stains, as does the non-senescent tumors. As you can see, we have the H&E stain, pan-CK stain, and an AR stain for both types of tumors, senescent and non-senescent. So there is an EH stain, which is the combination of two histological stains, a hemotoxylin and eosin stain, or I'm sorry, those are both the agents that are used. And this thing is the most widely used or golden standard stain that scientists use on biopsies when they want to look for cancer cells or if they suspect a reason of cancer. And so the eosin stains the extracellular matrix and the cytoplasm pink. So all the pinkish, I guess you could see here, would be the eosin stain, where you can, that would be the, like I said, the extracellular matrix, and the hemotoxylin stains the nuclei a purplish blue. So all the dark bluish dots that you see around here, all the little tiny like purplish dots would be nuclei stain. So this is important. They want to know, we want to be able to tell the difference between nuclei and extracellular matrix to be able to determine kind of like what are we looking at as the cell. And then we have the second stain containing a pan-CK staining, which is a strong and distinct stain of large epithelial cells. So with this stain, they're looking at epithelial cells, and then we have the AR stain, and that's looking at it's an androgen receptor, which is located on X chromosomes, and they're expressed in a large variety of range of tissues. They act as a protein that binds to male hormones. So cancer cells that are AR positive need androgens to grow. So these are the three stains, and we can see that for each set of tumors, we have mice that have PTEN genes turned on, mice that have both PTEN and TEMP1 genes turned on, mice that, on the other hand, that have PTEN and TRP53, and then finally the mice that have PTEN, TRP53, and TEMP1. And from this, we can see that the mice that have these two specific genes turned on, the PTEN and the TEMP1 under the senescent tumors, have huge mass in the middle, huge metastatic cancer slowly spreading around. So with these two genes on, the metastatic cancer is happening, coming fast. You could see it. Compared to the mice that doesn't have the TEMP1 gene on, it's just PTEN gene, that has no metastatic signs at all, no cancer signs. And so we could see from these two stains comparisons that the mice that have these two genes on under the senescent tumors are most likely to have metastasis. And the mice that have senescent tumors but don't have the TEMP1 gene active or has been deleted or deactivated, they have no metastatic cancer spreading or as fast as that. then we compare it to the, then we compare that to the non-senescent tumors. Both of these stains show, or I'm sorry, all three stains on both of these sets of mice show no metastatic cancer spreading at all. And we can see even here with the TIMP1 gene on, when it has been present, it still didn't show an effect or give an effect of metastatic cancer spreading. And that's partly due to the reason that it's under a non-senescent tumor cell. If we compare this, if we compare these two stains here, the pan-CK and the AR stain from both of these sets, the non-senescent tumors and the senescent tumors, we can see that it's the, I'm sorry, you can see that it's these two stains here that show the cancer spreading. And so we can easily see, right? So yeah, the TIMP1 gene plays a crucial role on whether or not the cancer is going to metastasize.
So this one is figure 1E. It's showing tumor cell division that is metastasizing. So in this figure, the dotted line is the barrier between the tumor, which is labeled epithelia, and the outside of the tumor. So once the cancer cells leave the inside of the tumor and are on the outside, they are metastasizing, which is not wanted. Each arrow is showing senescent cells, and each arrowhead is showing metastatic cells. So, for example, down here on the bottom left with P10 and TRP53, the metastatic cells have left the tumor and started metastasizing, which is why the red is on the outside of the dotted line. To the right, this graph shows the percent of metastatic cells that's close to the senescent cells. So they're extremely close for only TRP53, which is not a good sign because it means that the senescent cells are metastasizing at a quick rate if the percent has increased, and it's showing that TRP53 alone is not a good gene, once again, to have by itself.
And so move on to the next slide of conclusion. So the conclusions for I just threw some bullet points real quick. So clinicians are challenged by the metastasis of cancer cells. And this is partly due to the reason that it's a challenge, right? It's a challenge for clinicians due to the fact that there is very little understood about what happens at a genetic and cellular level. So we're still trying to figure it out. We're still trying to do research and, run experiments and try to see what genes work, what genes don't, what genes affect metastatic cancer spreading, and what genes help and inhibit it. And so better understanding of the mechanisms of how metastatic cancer is able to transition, right? And so by better understanding the mechanisms of metastatic cancer from a primary tumor would help scientists develop a better understanding for preventing it. So the more they understand, the better they'll be able to help prevent it in a clinical setting. There is still lack of evidence. So like I said before, there's still a lot we need to find out of how the senescent tumor cells are able to initiate this. So, the senescent tumors play a big role because they're kind of like what's going to determine whether these cells are going to start metastasizing or not. Given, which is kind of really odd because senescence kind of means, they're tumor inhibiting or cancer inhibiting. So very interesting. And so TIMP1 is labeled as a crucial regulator and for the initiation and promotion of metastasis. So by switching the senescence from tumor suppressive or to a tumor promoting response. So this guy here is like very important because, like I just said, it dictates whether or not the tumor is going to be suppressed or it's going to start metastasizing. And it is noted that when the TIMP1 gene is genetically deactivated, then the survival rate of the mice with senescent enhanced metastasis decreases. So when this thing is turned off with mice that have when it's turned off with the mice that have senescent enhanced metastasis, then their lifespan decreases. So it's very important to have that thing present. TIMP1 inactivation alone, though, didn't show signs of metastasis. It was just found that the TIMP1 loss was most likely associated with the process where it's called epigenetic silencing. I don't know, I don't have that on here. Sorry. But yeah, it's shown that TIMP1 most likely, or I'm sorry, that, yeah, it was found that the TIMP1 loss was most likely associated with a process that's called epigenetic silencing. And this is a process where expressions of genes are regulated through modifications in DNA or RNA. And so when the epigenetic silencing and the loss of PTEN happens, then yeah, patients get metastatic cancer, or the mice too. And so we move on to conclude that, patients that have low levels of PTEN and TIMP1 respond poorly. And they respond poorly to drugs needed to take for treatment in order to be able to fight prostate cancer. And the drug is called, I'm pronouncing it right, docetaxel. And yeah, so when patients don't have the PTEN or the TIMP1 gene present, then they respond poorly to the drugs needed to survive, and thus they have a shorter life, and longer, the disease is just free to go wild. And although the TIMP1 gene can be used as a biomarker to help scientists when treating therapeutic intervention, I'm sorry, not although, altogether, the TIMP1 gene can be used as a biomarker to help help scientists and clinicians when treating with therapeutic intervention. And so lastly, just for the critiques is what I have, some questions and critiques. We each had one. One of them was just a critique. Basically, one of my fellow classmates wanted to say that, if there was a better organized chart that was displaying the outcome of every combination of genes and its stains, then there would have been more of the stains would have made, as this type would have made more successful genes clear. So the genes that are successful in helping metastasis, inhibiting metastasis would have been a lot more clear to be visible or seen. Another one of my teammates had a question as far as if the TIMP53 is a good gene to have present, then why does it make cancer worse in scenarios with certain drug stains? Sorry, I keep doing that. And then finally, my question was, as far as figure 1D goes and the PANCK stain, so that showed a huge mass in the center, and you could see the metastasis spread, and that was present with the TIMP1 gene on. So I was wondering, my question was, if the TIMP1 gene acts as a regulator of initiation and promotion of metastasis, why does deleting it decrease the survival rate in mice that have senescent metastasis? And yeah, that's about it.